Hello, everyone, and welcome to Ask the Expert. We're very pleased to have Laura Jacobsen from the University of Florida with us today. Laura is the assistant professor and a pediatric endocrinologist at Florida. And as a physician scientist, Laura's research focuses on the understanding the role of the immune system in type 1 diabetes and improving clinical care and health outcomes for people living with T1D. A major facet of both of these is the conduct of immunotherapy clinical trials in those with T1D and individuals at risk for T1D. Laura has a special interest in mechanisms of action of these therapies, as well as biomarkers of efficacy and identification of clinical responders. Laura, I think you were here about almost a year ago exactly and talked to us about biomarkers. Isn't that right?
Yeah.
Yeah. Yeah. Laura's proud to serve as an investigator in TrialNet, TEDdy, NPOD, and the Type 1 Diabetes Exchange QI Collaborative. So thank you so much for presenting your work today, Laura. The title of her work is Early Metabolic Endpoints Identify Persistent Treatment Efficacy in Recent-Onset T1D Immunotherapy Trials. So go ahead and take it away. And if there's questions along the way, feel free to go ahead and put them in the chat box. We'll have hopefully a little time for questions at the end as well. Go ahead, Laura.
All right, perfect. Thank you so much, Alexis and Monica, for inviting me. I do love coming and talking with you guys. I feel like we have passions for the same types of research and advancing kind of these novel methodologies that I think are really great to get out to the rest of the field. And I've been very fortunate to work on this project with several members of TrialNet, which are listed here, and a great statistician, Dave Cuthbertson. So we were really excited to see what we can do about how we think about clinical trials, and are there ways we can do things differently from the status quo. So I'll be going over that. There we go. Here are my disclosures, just so everyone is aware. So what I thought I would start with is trying to look at the forest, or like the 30,000-foot view of what our goal is in type 1 diabetes immune therapies. And this is my opinion on what I think the kind of the big picture is that we're trying to identify immune therapies that either halt beta cell loss in people with early type 1 diabetes or delay that loss of beta cell um, over time, so that we can hopefully not only prolong what we call the honeymoon period when you have kind of a partial remission, because studies have shown that if you have a longer honeymoon period, you actually can have lower rates of complications like retinopathy and kidney disease, and even have lower risks of long-term hypoglycemia as well as acute hypoglycemia if you're able to even still make some insulin on your own, even if that is not able to take you off exogenous insulin. But I list here, that's a goal we would love for everyone who has type 1 diabetes, is to no longer have to be reliant on exogenous insulin from injections or from a pump. But I think also a big picture to think about when we're talking about immune therapies and the conduct of trials is that this hopefully could also apply to the really, really vital field of beta cell replacement for people who've had longstanding type 1 diabetes. And right now, people are really trying to find a way to hide those beta cells from the immune system. But, we might still need to be thinking about using immune therapies to prevent beta cell rejection and those kind of things. So I feel like you can learn a lot from these types of immune therapy trials, not only for people with recent onset type 1 diabetes. And what we want to do by having successful trials is we're benefiting people who are in those studies, but we want to move that to clinical care. We want people to be treated with type 1 diabetes like they're treated with rheumatoid arthritis or juvenile idiopathic arthritis. We want them to get diagnosed. We try and identify something about their specific disease that then we can treat with an immune therapy that modulates their disease. So that is the future, and we are getting closer to that. But at the same time, we don't want to forget what kind of the small picture is. We don't want to lose the forest for the trees. So in my opinion, there are still barriers to getting these disease modifying therapies, things that prolong beta cell health and halt the immune attack. There's still delays in getting these to successful review by the FDA or continued phase 3 trials, like kind of getting to closer to the endpoint here. And one of those barriers we see is that it takes a long time to go through each of our clinical trials, even in the new onset space. And that really, when we're trying different therapies with different combinations and things, it takes a long time. And if we can shorten that at all in the earlier phases, maybe that will allow us to test more therapies more quickly and can actually get them to patients in clinical care um, a little bit easier. So that is kind of the impetus for this study that we put together. But I still wanted to go over, well, why, if we have a successful way of doing clinical trials, why do you really need to talk about shortening clinical trials? And I'm not talking about shortening, phase 3 studies that would help us get approval for an immune therapy. What I'm talking about are early phase studies, so phase 2 or phase 1-2 immune therapy clinical trials, where another study is going to have to be done. We just want to know, is there a signal here? But if we're having to spend 5 years conducting a phase 2 study, that ends up being unsuccessful, that's a lot of time that could have been spent testing, 3 other therapies with a shorter outcome. If we can agree that we find a shorter intermediate outcome that is reliable and of value. Um, so not only on the right here, you can see how long it takes to get through the clinical trial process from phase 1 all the way to FDA approval. Uh, it's a decade or more. This is a long process. So anything we can do to shorten the earlier phases, I think would be valuable, especially when we're using repurposed drugs that have long safety records that we can pull from. I think that helps shorten that phase. But if we can not have to wait for a 1 or 2 year endpoint of a trial, maybe that could help us move from phase 2 to phase 3 faster. And then on the left here, since COVID started, there's been difficulty recruiting for clinical trials. It's just a different era that we're in now, where this is showing data from the GI field, but we're not able to recruit people as fast, as reliably, and it's hard for the entire field. So if we can find ways that these studies, patients are maybe not committed for as long, maybe ways that patients can all get a therapy, trying to remove as many of those barriers for participants as we can help them overcome some of the challenges that have occurred since COVID. So briefly, I want to remind everybody, I'm sure most of this audience knows what the standard endpoint for our type 1 diabetes immune therapy clinical trials is. So lots of great work has been done by Jerry Palmer and others looking at what is the best measure. So obviously we would love a metabolic measure that would show a great clinical benefit, right? So we'd love it to be hemoglobin A1C or reduce complications, but those things are just very difficult in the new onset period to show big differences, and they have a lot of heterogeneity within them. So we found over the decades a more reliable marker of beta cell preservation has been looking at C-peptide production. And a lot of that we can also see correlating with hemoglobin A1C or reduced exogenous insulin doses. But C-peptide area under the curve, specifically from a 2 or 4 hour mixed meal tolerance test, has been the standard clinical trial endpoint for the last 1 to 2 decades. So what I'm showing here is just from a paper by Mike Rickles that's just showing different mixed meal tolerance C-peptide curves. Just because I know when I started, area under the curve was a very foreign concept and I had no idea what people were talking about. But you can see that if you look at an individual person's C-peptide, it can be very different. And if you just look at, say, where they start at, there's not as much differences. The 2 hour value at 120 minutes, there's a bigger spread of differences, whether someone doesn't have diabetes or someone already has low C-peptide, as shown here in the blue lines. But the most reliable measure of C-peptide production has been the area under the curve. So I just shaded it in because it helps me visualize things that if you take this summed area under the curve from a mixed meal tolerance test at either following 1 year of treatment or at 2 years, that's been reliably found to find some therapies that have resulted in preservation of C-peptide as well as lower hemoglobin A1Cs in some studies. So this is great. It's been something that the scientific community has been working with the FDA on to show that this is a more than just a surrogate biomarker or surrogate endpoint, that this is something that actually correlates to improved clinical outcomes. And I bring it up here because we know this is a great measure, but are there potentially measures that are better, or are there measures that are better earlier in the clinical trial course? So instead of waiting 12 or 24 months, is there something that may be better to look at 3 or 6 months to give us a hint that something is having a treatment is having a positive effect? And speaking of types of treatments, I want to keep in mind, everyone to keep in mind, there are kind of two themes of immune therapies out there when we're talking about kind of immunomodulatory therapies. So we're not talking specifically about cell therapies or antigen-specific therapies. These are mostly just immune modulatory. They can be considered induction type therapies where they're given up at front in the first few days or in the first month, and then that effect can last for a while. Or they're therapy that can be given daily or weekly or monthly for an extended period of time, up to 1 to 2 years, the entire length of the trial at times. And those are going to have different kinetics. They're going to have different rates that they're affecting C-peptide production. So we just need to keep in mind when we're trying to say, is there an earlier endpoint? We need to get robust information to know if that applies to all types of therapies. So I just briefly wanted to go over a few trials here to say, why do we even think there would be an effect early on? So I'm showing 3 different immune therapies here. 2 are T-cell therapies, teplizumab and abatacept. So one is a anti-CD3 monoclonal antibody, and the other inhibits T-cell activation or co-stimulation. And then the one on the bottom in yellow, golimumab, is a cytokine inhibitor, so an anti-TNF antibody. These are all trials in new-onset type 1 diabetes and all looking at C-peptide. It's area under the curve, and the one on the bottom is actually just showing the change from baseline as opposed to the absolute values. But what I want you guys to notice here is that we clearly at 12 and 24 months are seeing separation between the treated and placebo groups. But if we try and focus a little bit sooner than 12 or 24 months, so this is looking at the teplizumab trial. So we can actually see at the first dots there are here at 3 months and 6 months. Oh no, sorry, this is 6 months. So you can already see this separation. And not only can you see separation in these values, but you can also see that their trajectories to get there are very different. So there's a pretty characteristic decline in placebo treated individuals. Their C-peptide is the natural history of the disease, just slowly decreases over time. It's a little more rapid in younger people. It's a little slower in older individuals. But you can really notice a difference in the first 6 months. But not all drugs are the same. So this is abatacept, which is more of what I would consider a maintenance therapy. It's given every 2 weeks for an entire 2 years of this trial. But you do see a difference in the rate of fall in the first 3 and the first 6 months here. That even though this is a maintenance therapy and they've only been on it for 3 or 6 months, may help predict what their 12-month outcome could be. So there are definitely hints of this in the trials that we've already conducted. And then the same thing for golimumab. You can definitely see here that during the year of treatment, the treated individuals had basically no decline in their C-peptide, whereas the placebo individuals had a very predictable fall in their C-peptide. And you can see that very early. I don't want to generalize, though, and say all trials are necessarily like this because placebo groups can be different. So if we look at the anti-thymocyte globulin TN19 trial done in TrialNet that compared ATG plus placebo, or sorry, ATG plus GCSF, or a placebo group in blue, green, or red here, you can see that the placebo group up in the first 3 months here actually had a very slow rate of fall. So this is a group that was making a little more C-peptide on their own, may have been a little bit older than other trials. And so their characteristic decline in C-peptide didn't happen until the 3 to 6 month period. So we want to keep those things in mind when we're thinking about earlier endpoints if we want to look at 3 months or 6 months. I don't do as much, you won't hear me talk about the 9-month time point much because in the data we looked at, not all trials included data from mixed meal tolerance tests at 9 months. So we did not analyze that information. And then finally, there are some trials where you see it. This is baricitinib, JAK inhibitor. This trial was done in Australia. And you see the characteristic decline in C-peptide in the placebo group. Again, this is C-peptide AUC, or area under the curve. And you see what we see in some trials is an increase in the C-peptide in the first 3 and 6 months. So those are very different responses, and you can clearly tell that these are showing a benefit from this therapy. And if we analyze that data, could we predict that this, 3 or 6 month change readily or reliably identified the 12-month endpoint? So that's what we.
Laura, was that one changed from baseline? Do you know?
No, I think this is absolute.
Absolute? Okay, thank you.
Yeah, no problem. Mark, do you have a question? I see your hand raised.
Yeah, just again, maybe you agree, disagree, Laura. It's important to note that these are means and their distribution, and that individuals within these groups can vary. Meaning, it looks really this picture here that you rise or whatever, but their experience, and it's hard to tell at an individual. Yeah, that's all I wanted to say. Do you agree with that?
So I agree with that, Mark. So definitely, there are limitations to looking at all subjects and grouping them together into an average. I do think there's great benefit in looking at individual responses, and I think that is a fascinating topic. And I'm actually a huge fan of variables like the quantitative response QR variable. This is just it's still looking at mean measures when I'm talking about earlier endpoints, because we just wanted to know if our classic endpoint could be predicted by an earlier time point, not individualizing the response yet. So I think that's a great point that we want to keep in mind. So if you had to kind of visualize, I haven't seen the individual data for baricitinib here, but there are going to be some people who had a really robust response up. There are going to be some people who received baricitinib who may look just like the placebo group. But there is enough of the participants who received baricitinib that the average is able to be pulled up like this. But I agree, to benefit individual participants, individual responses, and how that person does compared to other people of the same age and starting C-peptide is of the most value. I agree.
Laura, can I just ask on this trial and the previous slide, those trials, did they all have the same numbers of participants, or was it really wildly variable?
I don't know off the top of my head. I bet I think they ranged anywhere from like 60 to 100 participants total. If I just can't, I don't know the exact numbers. So
Yeah, just
they're
random,
not
just
teeny
ballpark.
tiny.
Yeah, it's pretty small. Okay, thanks.
Mhm. All right. And then just to give people an understanding of why we chose some of the metabolic markers we chose to look at earlier time points, in addition to the classic C-peptide endpoints, is we've done a lot of work in the at-risk population, so people who have multiple autoantibodies and are at high risk of developing type 1 diabetes. And we've tried to look at markers in addition to just glucose or just C-peptide in that group to see what could reliably identify high-risk participants. So this is a figure from a paper by Brandon Nathan and Jay Sasenko and others, where we're looking at the risk of developing type 1 diabetes for people who are multiple antibody positive. And you can see here, if we look at the green and the black lines, those are people who who are only using a abnormal 2-hour glucose from an OGTT to identify their risk of progressing to type 1. Or people who are using what we call an index 60, a very specific measure that incorporates both C-peptide and glucose into the same metric. So it's developed by a logistic regression using DPT-1 data. And if you have a low reading for that value, or a low reading for the 2-hour glucose, you have a low risk of progressing. But if you have a high reading for the 2-hour glucose, what we classically think of as a high risk of progressing, it's still not very reliable at saying you will or will not get type 1. Whereas if you actually use that combination measure that includes glucose and C-peptide, what we call the index 60, that is much more able to identify a high risk population. So we've just been really impressed by using these combination measures as opposed to a C-peptide or a glucose alone. So that is what spurred us to say, hey, can we look at this in mixed meal tolerance tests? Because we know glucose is affected and improved in people with type 1 who've received immune therapies, and we know C-peptide can be improved, and both of those together may be more valuable than either individually. So that's how we came up with other our list of variables that we tested. And you'll see in the paper, I kind of have all the figures, you know, kind of one after another about, hey, here's how we see differences at early endpoints. But how it naturally evolved actually is we wanted to look is, do we see a difference when we're combining C-peptide and glucose? Do we see early effects with these, what we think are really reliable high-risk biomarkers? So we actually can visualize that in a really neat way that I know can be very complicated, so I'm going to try and walk through it. But Jacobsen has developed these glucose and C-peptide response curves where you're plotting the glucose values from a mixed meal tolerance test on the y-axis and the C-peptide values from a mixed meal tolerance test on the x-axis. And basically at the 30-minute time point, the average C-peptide and glucose are a specific coordinate. And then at the 60-minute time point, 90-minute time point, and 120 minutes, we exclude the fasting sample because it's markedly different. And with this, it's a really nice way to show how the actual curves move with time. So you would expect if someone here in the dashed line has this shape of curve, if you plotted their glucose and their C-peptide at the time they enroll in a trial, if they're in the placebo group and they are not getting an effective immune therapy, we would expect their glucose to rise, which is to go up on this curve, and their C-peptide to go to the left. And so the entire curve here shifts. So this is just a nice way to visualize it as opposed to just looking at data. So we did this for the TrialNet new onset studies to say, hey, do we even visualize the data in these trials we want to analyze? So we took 6 different trials in TrialNet. 3 were successful and 3 were unsuccessful. And there was a second arm from one of the studies that was also unsuccessful. So we've got pretty big ends here. So 252 participants were in successful clinical trials and 361 participants were in unsuccessful trials. And if you look at the bottom here, this is the 12-month endpoint. We're showing here in the red dashed line, people who were in the placebo groups for these successful studies and people in the blue dashed line who were in the treated groups. They all start out at about the same C-peptide and glucose values for their mixed meal tolerance test. But you can see by a year, the people in the placebo group are doing much worse. They have much higher glucose and much lower C-peptide. And you can visualize both of those metrics in the same figure. Whereas if you look at the blue solid line, those who got the treatment, their C-peptide is improved and their glucose is improved compared to the placebo group. So you could say, okay, great. You can visualize the difference between treated and placebo. What about in the unsuccessful trials? So the unsuccessful trials are here on the right, and you can see those who got a treatment in blue overlap at 1 year with those who got placebo. So great, this visualization is working. What happens if now we want to look at earlier time points? So we did the same thing at 6 months. And if we keep looking at the right here, the unsuccessful studies, you see at 6 months, there's complete overlap between the MMTT, glucose and C-peptide values for the treated and the placebo. But here in the successful treatments, you're already seeing an improvement in that C-peptide and glucose values. And so what about 3 months? This is getting a pretty tight graph here. So at 3 months, if we look at the unsuccessful treated participants in blue here, so they moved up and to the left by a certain amount, almost similar, almost exactly the same as what the placebo participants did. So their glucose rose and their C-peptide fell in a very similar fashion. Whereas at 3 months, these successfully treated participants had almost no change in their glucose and C-peptide. So we did see in these trials we wanted to study that 3 and 6 months, you did notice a difference in glucose and C-peptide measures. So we just thought that was very encouraging for us to start our analysis. So what was our analysis? So we wanted to determine if you could see a treatment effect. So was there a statistical difference between the treated group and the placebo group at 3 or at 6 months? And could you see that in the successful studies, but not in the unsuccessful studies? Again, these are all new onset type 1 diabetes, people within 100 days of diagnosis from type 1 diabetes. After we determine, can we see a successful treatment effect at an earlier time point? We want to know, does that earlier time point predict the 12-month time point, kind of the classic endpoint? Because if we find an earlier endpoint that shows a difference, but it doesn't predict what's going to happen down the road, that's less hopeful when we're trying to replace it in an earlier phase study, right? That would make us concerned that maybe this is not reliable for carrying forward to future trials. And then the final thing is, if we can do a study in only 3 or 6 months to find an effective or not effective therapy, is it going to be a sample size that actually makes it practical to do? So we decided that the only sample size that was practical was one that was smaller than the current trials that were being run. So set a pretty high bar for that. So this is the analysis that we did. So on the left, you'll see what trials we included. So basically, we took all of the new onset studies within TrialNet and in a post hoc manner went back and looked at these earlier time points. So we had 3 successful trials. So that was people with new onset diabetes treated with rituximab, people treated with abatacept or low dose anti-thymocyclobulin. And then there were 4 treatment arms that were not successful within TrialNet. So those are people who received mycophenolate mofetil and daclizumab, GAD, antigen-specific therapy, canakinumab, which I believe is an IL-1, anti-IL-1, and ATG plus GCSF. That arm was not successful at meeting its primary endpoint. So what we did is we looked at the classic 12 or 24-month time point, and then we said, what do we see if we look at 3 or 6 months? And not only are we going to look at C-peptide area under the curve values, we're going to look at measures that include only glucose, and then we're going to look at what we're most interested in, which were measures that include combinations of glucose and C-peptide. And not only were we going to look at the absolute difference between those time points, but we wanted to look at the change from baseline, because even though there may be a small difference between 2 points, they may have very different slopes that may actually have more power to them than looking at absolute values. So that was the analysis that we planned. This is just a summary of the components of the trials. So you can see here, Monica, the numbers for each arm. So it's definitely, I mean, each arm had at least, 22, usually in the 30s, and then the treatment arms often were 2 to 1. So while the sample sizes are not huge in the scale of clinical trials, they are not pilot data. And I'm only listing here the administration frequency for the successful trials because that's what I'll focus on. But rituximab and anti-thymocyclobulin are what I would consider induction agents. They were given over a couple days to 1 month of the beginning of the study. And then the outcomes were looked at a year, whereas abatacept was given monthly after and kind of a ramping up phase at the beginning for 2 years. And the 2-year endpoint was what was the primary endpoint. So those 3 trials were statistically significant in their area under the curve comparing placebo and treatment, and the other ones in the bottom half were not. So just to show you the data we included. All right, so this is just a schematic to try and help you, really, it was to help me visualize the difference between looking at absolute differences in C-peptide and the change in differences or the deltas in C-peptide. Because I've always been used to seeing the absolute difference. So if we look at, say, 3 or 6 months, you just look at the 6-month mean AUC peptide minus the placebo AUC peptide. That difference is what you get. That's what you're looking based on the variability. Is that statistically different treatment from placebo? So that's classically what's done at 12 or 24 months. And so we went ahead and took the classic measure. When we talk about C-peptide area under the curve, the actual mathematical formula that's used is actually the natural log of the C-peptide area under the curve plus 1. I think that's just for transformational purposes. And so we took that classic measure and we looked at 3 and 6 months, and there just wasn't a big enough difference in the absolute value between treated and placebo. Okay, we weren't really expecting that. Like the lines are very close together at those points. And again, this is just a schematic representation that I made where the treated group is in blue and the placebo group is in red, and the duration of the trial in months is along the x-axis. But then we said, well, let's look at the difference between the treated and placebo if we look at the first 3 months of the trial versus the first 6 months of the trial. So to me, I had a hard time conceptualizing this. So when I can't conceptualize something, I make a figure about it. So basically, I'm going to take the difference between time point at 6 months and the time point at 0 for the mean AUCC peptides in the treated group. And then I'm going to take the difference in the placebo group from the 6-month time point minus the 0-month time point. And it's that difference that I'm looking at. So not only is it taking into account some of the absolute value, but it's taking into account how different the change is from each other. And we're calling that kind of more of a dynamic measure over time, as opposed to the classic absolute difference that is used in typical new onset trials. So we were really actually excited to see that if you looked at the classic AUCC peptide, but you took the delta, the dynamic measure over 3 or 6 months, you could find a statistical difference when looking at those successful trials. So rituximab, abatacept, and low-dose antithymocyte globulin, if you aggregated those together, you could see a statistical difference between the treated and placebo group. So you can identify a treatment effect. So that was nice because people are very used to this measure. So it's always hard when we try and introduce new measures to people. It's change is hard. So that was positive. So that's great. And then this is just another way to, to visualize that difference. So what I'm showing here is the AUCC peptide in the treated group in orange and the placebo group. Again, this is the mean AUCC peptide over 3, 6, 12 months. And then I'm going to show you in bar graphs here what the differences are. So if you just look here where the mouse is, the difference between the absolute 3-month value and the absolute placebo value at 3 months, there's no difference. But even though they're so close together, the rate of change is different. So that that's why you can see a effect so early. So it was very encouraging to see at 3 and 6 months. If you looked at the delta, the change from baseline, you do see a difference using the C-peptide AUC. And obviously, when you get out to 12 or 24 months, you see a difference not only in the absolute values, but also in the dynamic values. So this was just me trying to visualize it in different ways, kind of to help myself understand it. So after doing the kind of the classic C-peptide AUC, we wanted to take our knowledge of other measures you could derive from a mixed meal tolerance test, and we used our knowledge about combination measures in the at-risk population. So I talked about index 60. That's that combination measure that includes glucose and C-peptide. If anyone wants the details, it includes the fasting C-peptide and then the 60-minute glucose and 60-minute C-peptide from the mixed meal tolerance test. So you'll see here we have a long list of C-peptide only measures, glucose only measures, and then a couple combined measures that we wanted to see, are any of these more specific for identifying a difference between treated and placebo at an earlier time point. You know, there's definitely a lot of glucose changes that are going on shortly after diagnosis. So if you add that into a measure that includes C-peptide, might that give you more detail than a C-peptide alone, which may not change much in a 3-month period in anybody, right? I don't think it's falling at that fast of rate in the general trial population. And then Hiba Ismail, who was the senior author on this paper, also brought up a great point, is maybe we're having more influence from first phase insulin release or second phase insulin release. So we really wanted to look at different fractions of the mixed meal tolerance test and early versus late responses to say, is there something physiologically that is able to be detected earlier instead of at a later time point when we're kind of just looking at the whole area under the curve. All right, so this is kind of the main data slide, results that I wanted to go over for this. And what I'm showing here are how the different measures that were on the previous slide compare to each other. So it's really tough to try and compare these different measures in a statistically sound way. So we worked with Dave Cuthbertson and we actually used what's called the F statistic, which kind of helps you rank the strength of the ability to differentiate treatment effect with these different measures. So all of the measures at a certain time point were analyzed together, and then those with the highest F value or F statistic were the strongest in their ability to differentiate that risk or that treatment effect. So all of the measures actually a lot of them had a statistical difference and were able to detect a treatment effect, but we wanted to know what is best at detecting a treatment effect at which time point. So you'll notice here what we're showing along the x-axis is the time points of the trial. So we'll focus on 3 months, 6 months, 12 months, and 24 months. And then along the y-axis is actually this statistical value called the F value that is showing us how strong the ability is to detect a treatment effect is for each of these markers. So if we look at, we'll zoom in a little bit here. If we just focus at 3 months, all right, so we're looking at the 3-month time point, and we can see here these 3 values in the first column are either 120-minute C-peptide area under the curve, or the classic transformed C-peptide area under the curve. Those are all have relatively low ability at 3 months to detect a treatment effect. Whereas if we go to the second column here at 3 months, these are looking at our combination measures. So if you look here at the top, this yellow color, this is the centroid ratio, which is another combination measure that uses the glucose and C-peptide response curves that I showed on a previous slide. And you can see that that value actually had a really good predictive ability to identify a treatment effect. So I put a dashed line here around a F statistic of 12 just to kind of compare across graphs. And then other glucose-only measures, metabolic measures, actually show differences between treated and placebo groups when we're looking at the successful trials within TrialNet here. So early on, glucose measures and combination measures are the most effective at differentiating treated from placebo. But that's not true once we go out to 6 months. So if we look at 6 months here, we can see that the classic transformed C-peptide area in the curve, it actually has the strongest discriminative ability to identify treated from placebo. Again, these are all dynamic measures. So once we realized that the change from baseline was much more valuable than the static measure at a single time point, we did our future analyses with just the dynamic measures. So over the first 6 months of a trial, the change in the standard C-peptide area under the curve has the best ability, because it's got the highest F value at 6 months, to detect a treatment effect between treated and placebo. The next highest measure was in red here. This is the AUC ratio. So this is the AUC of the AUC C-peptide over the AUC glucose. So that is another measure that's including both glucose and C-peptide. So we're really excited to see that, but actually it didn't perform the best. The classic C-peptide area under the curve performed the best. And then if we look out to 12 months here, you can see there's nothing that surpasses the C-peptide, because again, that's what we're considering a successful trial. And then the same thing at 24 months. The later you are in a trial, the less glucose values have an ability to predict a difference between treated and placebo. So this was really encouraging. We realized we could probably just use our classic measure, but over as a change from baseline to detect an early treatment effect, and that it would also be of interest maybe to use this combination measure to see how reliable it was. So once we do that, then we need to decide, okay, these measures that we're looking at, are they actually able to predict that 12-month or a long-term outcome? So what we did is we looked at the R squared values for each of our metabolic measures at 3 and at 6 months to say, are we getting a good ability to predict the variance in a measure that predicts our 12-month endpoint. So to kind of put that more simply, if we look here, the C-peptide AUC or the AUC ratio at 3 months, they have okay ability to predict the 12-month C-peptide AUC. That's what we're trying to predict. And we did adjust these for age and baseline C-peptide because the variability is very high if you don't do that. So here you can say, well, these two measures can have okay predictive ability, right? We're trying to get to a R squared of 1 is the best, and R squared of 0.5 is like flipping a coin. But let's look at 6 months for our measures here. So I circled kind of the top 2 measures, which doesn't include our AUC ratio. So the top predictive measures at 6 months were the change in C-peptide AUC or just the 120-minute value. The AUC ratio here is 0.68, so it does okay, like a pretty reasonable predictive ability for these measures to predict the 12-month endpoint. But even if we can predict the 12-month endpoint, is this going to be feasible? So let's look at kind of these top measures here and what a sample size would look like if we designed a trial to detect a difference at 3 or 6 months. So here we're looking at the change in C-peptide over 3 months. And if I'll circle it here for you. So the change in C-peptide, if we want to say what treatment effect are we looking for, right? We're going to sit here today and we're going to design a trial. What's the effect size that I'm going to look for? Well, if I tell you that the rate of fall of the C-peptide in the placebo group is 0.1, right? So say this falls 0.1 nanomoles per liter. Well, then I want to know how much better than that fall I can do to try and determine what my effect size is going to be. So one thing I want you to look at, if I say, we're going to do 0.1, we're trying to get better than a 0.1 fall. If I do 50% of that, if I'm going to fall only by 0.05 in my treatment group, that's going to give me sample sizes at 3 months per arm in the hundreds and hundreds, just untenable. You can't do this. So let's say we might see a really robust treatment effect at 3 months. If I think the placebo is going to fall by 0.1, I think that the treatment group is not going to fall at all. So it's going to be 100% better. If I think that these are still incredibly large sample sizes to have 115 participants per treatment arm, so in the treated group and in the placebo group, those trials are just completely unpractical to do. So it's kind of looking like even though 3 months may identify a treatment effect, that they are not going to have the best predictive ability, and these trials are just not practical to do with the sample sizes. So what if we looked at how much the C-peptide falls in the placebo group over 6 months? And what if I think it falls by 0.2? All right, well, maybe at 6 months, I think we can do 100% better than that. And we're not going to have any fall at 6 months. So in that case, with our 2 kind of top measures, that would only require a sample size of about 54 participants, which is smaller than most of the trials that were conducted with a 12 or 24-month endpoint. So this is not a completely unreasonable effect size. So this effect size was seen, if you say we're going to do 100% better than the placebo group at 6 months, that was seen in the TN19 ATG arm. It was seen in the golimumab study, and it was seen in baricitinib, the JAK inhibitor study. So we see that in our really successful studies, that there is a 100% improvement over placebo in that shorter time window. Not over the long term, because over the long term, the treatment group will eventually start to fall parallel. But at the beginning, there is kind of sustained or improved C-peptide production that could make these small sample sizes actually realistic. So as I kind of wrap up here, I just want to go over some of the limitations of this analysis. Again, we're combining different treatment effects. So I've got 2 kind of induction therapies, and I've got 1 maintenance therapy, and we've kind of grouped them all together in the aggregate. We did pull them out individually and look at each of the trials, and a lot of the data is driven by the TN19 and the abatacept data, because they have the strongest effect, whereas rituximab just it's significant, but it's kind of the least significant of the significant trials, if that makes sense. So we definitely need more data on different trial treatment courses, if they're maintenance or induction. I would love to be able to look at if it's a T cell or B cell therapy. Is there something about the timing of a treatment effect that we could identify? And we did not adjust for multiple testing here. We have not yet validated this in an external data set. And people often ask us, well, what is the FDA going to say about using a 6-month time point? And I agree, we need a lot of data on that. But my goal for such a time point would actually be for these early phase 1, phase 2 trials that then would lead to the conduct of a big phase 3 trial using a classic accepted endpoint by the FDA. So this is not something I would expect would be an FDA approving endpoint at this time. So you could say, Laura, why are you trying to shorten trials? Like, trials are fine. We don't think they take that long. Well, I think participants would agree if they're in the placebo group of a trial for 2 years, that's a long time. And people have been looking at ways to improve trial design, not only in type 1 diabetes, but in other spaces. And there's really been a big push recently because we can find the same effective data and have better clinical trial designs that give more patients more benefit, and that give us answers more quickly. So I'm kind of putting up here a lot of recent papers that came out kind of supporting this, that we need to be thinking about new trial designs, and finding intermediate endpoints such as this may help us design those. So whether we want to stop a trial early if they don't reach an intermediate endpoint, whether we want to adapt a trial and switch to a different therapy if it's not being effective, all of those, I think now is the time for us to experiment with that and validate some of these endpoints for such a trial. So what my summary is that when we're looking at endpoints in T1D immune therapy trials, the change in an endpoint is more valuable than the absolute measure. 3 months is probably too short of a time with the degree of variability and the large sample size that would be required. So if we were going to have an intermediate endpoint on which to make a decision about a therapy, 6 months is probably the shortest time that you could do that in, and still predict reliably that 12-month endpoint of interest. And the top measure for that is our classic C-peptide area under the curve. But also, I think it would be great to explore more the AUC ratio and other combination measures that also were up there. And then we wanted to let everyone know that we are actually in the process of validating this methodology within the Immune Tolerance Network. So they have 3 or 4 new onset clinical trials using different therapies that also have mixed meal tolerance data. So we're going to see if 6 months using one of these 2 endpoints or others is seen in that data set, which would really allow us then to move forward with designing a trial to prospectively test this. And then, as Mark brought up earlier, I think that trials should be designed not only with these metabolic endpoints that are kind of used as means, but also with individual responder endpoints. But that is a separate talk, and I would love to talk about that as well. And also the group out at the University of Washington or at Benaroya Research Institute and Jeff Krischer and all of them have worked really hard to develop a responder signature for individual participants. So I just want to leave everyone with this figure. So this is from the juvenile idiopathic arthritis literature, and it is showing over the last 5 decades their approval of immune therapies in pediatric, so juvenile idiopathic arthritis. So they have had 7 immune therapies approved for a pediatric indication that they can now use for different phenotypes, different endotypes, that kind of thing. So we have a long way to go, but we can get there. This is possible, and we need to be thinking about type 1 diabetes the way they thought about juvenile idiopathic arthritis, so that we can get our immune therapies to patients so they can actually get benefit at the beginning. So I kind of posit that maybe if we go back to our 30,000-foot view, we're excited now that we can put teplizumab on this map, but we need to move other therapies through to FDA approval, not only in the at-risk, but in new onsets. So that is it. I'm happy to take questions.
Thanks, Laura. That was a really beautiful talk. I like how you organized and tweezed it apart and explained things. That was really helpful. I have one question to start us, and everyone else feel free to, turn on your camera or unmute and ask questions. But I wanted to start with this question. I understand the need to validate this, to see how those short-term measures, whether they predict efficacy at 12 months and 24 months. I understand that, but I'm wondering if we flip that around and say, let's look at these for predictors of treatment failure or failure to response to the treatment early on. It seems like you don't need to validate that to me, because there is not a reversal. It's across the board. We know if that C-peptide is starting to drop, even with the other variables, can't we say with certainty that those subjects are not responding to the treatment? And
So I see what you're saying, right? So the tough thing is that how do you know how much C-peptide drop is enough that it's going to actually be considered for that person enough? So I think we've talked about this with the quantitative response variable. If you can incorporate the age of the person in the starting C-peptide, I think that's great. Because you can predict, like, you should only be falling at this rate. And if you're falling more than that, then this is not working. Or if you're not, if the drug is not improving that any. So I totally agree. That's more going to be a responder difference if we can all as a field agree that those two variables in that model of decline are enough to say this is effective or not. Does that help?
mm-hmm.
I had a quick question. And this might be a little bit of a big question. Can we just talk a little bit about the C-peptide? How standardized is this metric, and how many labs sort of measure it in the US? And do you think they all use the same standard? Is it standardized, or is there room for improvement?
Yeah, so it's a great question. So all of these C-peptides were run within TrialNet, so they were all run within a central laboratory who does them all together. So they actually are very well standardized and validated, and they have. I was actually talking to Dr. Winter, who used to run the core lab for TrialNet, and they have 4 different standard values that they QC it against. And within the TrialNet C-peptide assay, the coefficient of variation is only 3 to 5%. So I think within a research setting, you can say that that's a very reliable measure. When you start going to commercial labs, I think that it probably is going to be wider. So when you talk about clinical applicability, but I think right now we just have to get something approved to be used in a clinical setting. So, I think we'll use the most reliable data we have, which is the kind of TrialNet C-peptides.
Well, that's good news. The TrialNet is got that covered. And then I just wanted you to sort of think or share with us, what are your thoughts on how we can close the gap between patient participation? This is a big issue in trials, and the pediatric engagement, to recruit and follow patients through this process, sort of like shepherd them through. Like, what is it going to take to get these people, in the TrialNet, realm?
Yeah, so I think a lot of kids, families with young kids are more eager than teenagers and adults to participate, but the drug trials are not often open to young ages. So trying to get drugs that are going to be tested in adults to go ahead and open to young children, because that's a group that you'll be able to, see the outcome sooner, because they're going to lose that C-peptide quickly. You're not going to have to wait as long for, like Alexis said, like a loss of C-peptide, or to realize that they're not responding. So I think trying to get those open to younger ages is going to be the big thing. And then I think we need to ask participants, right, with like community advisory groups, like what is the barrier now as opposed to pre-COVID for participating? Because it's hard to know.
Mm. Yeah, well, teplizumab is 8 and up, right? Now?
It's 8 and up, yeah. So it's 8 and up, and I think that's a difficulty for clinical trials in the prevention space, right? Because some people may say, well, I can get, if I'm 8 and older, I can get teplizumab if I'm at high risk for type 1. You know, why would I do a trial that has a placebo group? So I think that's another reason we have to think about redesigning trials. There's not a therapy approved in the new onset space yet. Hopefully there will be. In which case, you know, having placebo groups is probably a big barrier for participants to want to enter.
Yeah. And you wonder if you can target the whole teen group, you know, that
Yes.
cohort, you know, if you can really get them on board and prove up over there, then you could
Yeah.
maybe pull down into the pediatric cases.
Yeah, and I don't know if like because they're doing more like remote learning or if it's they have too many sports, you know, they have a lot of things going on in their life. But yeah, that's a group that would be great to treat.
Yeah, I
And
was going to ask Laura, when you brought up the fact that since COVID across the board, I mean, I think you put up something from gastroenterology, but
Mhm.
you said across the board, people are having trouble recruiting. What do you think that is, or what have you heard from other groups, but specifically for diabetes? What do you think is causing that decrease since COVID?
I could speculate, but I honestly think we probably need people to be doing some more qualitative studies on this. I don't know if people have a different risk threshold now that COVID is out and we're talking about immune therapies. I don't know if people are having more monetary concerns and, you know, taking off work and doing this extra, having this extra burden is still
Yeah.
is too much now.
Yeah.
Yeah, I think we need to ask more people too. I'm
You know,
just speculating.
the FDA has been focusing on decentralizing, so anything that can decrease the burden and the number of visits for the patient.
Yeah.
And I think that's challenging when you have something like mixed meal tests that you want to do occasionally. One more question about the data that you presented. Was when you do the standard mixed meal test 2-hour or 4-hour, is there a record of the C-peptide at time 0?
Yes, we
So
collect it. We don't know
essentially
it.
a fasting?
Yeah, yep. So we have that data again during the trial. We don't know it. We're usually blinded to it, but yeah,
Yeah.
we collect it at 0 and then every other time point. Yeah.
And you compared that along with the other measures just to see?
I'm trying to think. I'm not sure if we looked at fasting because historically we've not been able to see enough of a difference that it hasn't typically even made our radar because of how ineffective it is. But I'm sure like I will happily see if our statistician can run that, but I have low hopes.
Yeah, I wonder with the increased sensitive, the ultra sensitive test for C-peptide now, and if you're looking at a percentage change, I'm just curious what that, because that certainly would be much easier. And the data I've seen, like from Europe, where they did a little more collection of the fasting, it does mirror the changes that you see during the stimulation test. I just wonder if it's been difficult to detect because they're so much lower to start with. It'd be interesting to look at.
Yeah. We can definitely look at it. Yeah.
Yeah.
It would be nice. It's just not been as a robust a measure, but yeah.
Yeah.
I have one last question. If you're talking about these other data sets, Enodia, India, and Australia, TrialNet has, and I'm sure Canada has some data. So what if, is there a possibility to bring all these data sets together and in some way, combine their analyses? I know there, it's all different sort of flavors of data collection and test C-peptide standardization, everything. But has anyone talked about that?
So I think it would be great to have some kind of like repository of data like that. And NIH trials do get raw data uploaded to the NIDDK repository. It's tough when you go, especially outside the country, like, are they collecting the same time points in the same way that's comparable? So I think definitely within, like, we're going to look at the immune tolerance network, which uses the same assays and the same time points for the mixed meal tolerance test. Beyond that, it might be hard to do data cleaning to match everything, but I think it's definitely worth looking. We just haven't looked yet.
Okay.
All
More,
right.
more things to do.
I know.
Any other questions from the audience? Well, Laura, thank
Oh,
you so
sorry.
much. You got one?
No, that was
Okay.
marked before.
Okay. Thank you again, Laura, for presenting this today, and thank you for all the work that you're doing for patient care and for, especially in this clinical trial space, looking at endpoints and transformation of the clinical trials for type 1 diabetes. We really appreciate your work and can't wait to hear more about how this analysis kind of, the validation process for this analysis.
Yeah, thank you guys so much. I greatly appreciate the opportunity to share it.
Thanks for being a
Thank you.
T1D trials champion.
Thanks.
Bye.
Bye,
Bye.
everyone.